Good morning, everybody. And I would like to thank the organizers to again invite me to this annual meeting, which is always very interesting. I think here it is. Okay. So as Suman said, my topic today is to talk about extrapolation in the context of biosimilars. So the concerns that have been frequently voiced about biosimilars is that they may be of low quality, that the safety database may not be sufficient at the time of licensing, that they may have increased immunogenicity, or even that efficacy may be different from the reference product. But the most contentious issue really for physicians and patients is the issue of extrapolation of data, which means the licensing and the use of biosimilars in indications that have not formally been studied for the biosimilar. And you probably know many of these publications from Learned Society discouraging the use of biosimilars in extrapolated indications. However, I have to tell you that extrapolation of data is not a new concept. It's very well known in the context of biotech medicines, and it is based on the totality of the evidence to demonstrate comparability of different versions of the active substance of a biological. And when does this occur? Well, the scenario that we know very well already is the first one, change in the manufacturing process of a given product. And here we compare the pre and post change product from the same manufacturer. And you all know that we have an ICH guideline that has to be followed in this context. So the second scenario, which is newer, is a biosimilarity exercise. And here we compare two versions of an active substance from different manufacturers, but the principles are the same. And just to show you the extent of the comparability exercise depends on the type of change and also on the extent of differences we observe. If the change does not have an impact on specifications of the product, or if there are changes in the specification, but we know they do not impact safety or efficacy, physicochemical and biological testing is sufficient. However, if an impact on safety or efficacy is possible, additional data have to be provided. And this does not necessarily mean clinical data. And the reason is because I would like to remind you that the sensitivity to detect differences between two versions of an active substance is highest with analytical testing, with state-of-the-art analytical methods, followed by functional assays. And the most blunt tool we have actually are the clinical studies. They are just there to confirm biosimilarity. And sometimes we might not even need them anymore, as was previously highlighted by the previous speaker, Peter Richardson. So having that in mind, I think this is very important for the following discussion. What has been observed is that changes in the manufacturing process actually can change quality attributes, and with the sensitive methods, you can see them, you can make them obvious. And Schiestel and colleagues have followed several originator biologicals on the market. They have analyzed different batches and what they saw. I think this is for a rituximab, for the originator rituximab, that there was obviously a change in the manufacturing process around in 2010, which led to an increase in the afucosylated species, and following this, an increase in ADCC activity. So such changes do occur. I don't say that they occur all the time, but such changes do occur in the context of a change in the manufacturing process. And changes in manufacturing process of biologicals occur frequently. This is not a rarity, this is the rule. And as you can see on this graph, for Remicade, we have seen more than 35 changes in manufacturing process since licensing. And although these probably were mostly minor changes, I think it's fair to say that the product currently on the market is not identical to the product that has been licensed several years ago. And in this recent paper, the clinical experts from the Biosimilar Working Party wrote, we also highlight that extrapolation of data is an established scientific and regulatory principle, and that in the context of major changes in the manufacturing process, so that's a scenario where we usually want to see some clinical data, we are not aware that there was any case where additional clinical studies with the changed product in additional or even in all approved indications have been provided or have been requested by the regulator. What are the underlying considerations for extrapolation? Usually, we consider extrapolation unproblematic when the mechanism of action or the receptor involved in the extrapolated indication is the same, and if there are no unique safety concerns, especially with regard to immunogenicity. If the effect is mediated by the same receptor, but there are target cell-specific difference in downstream signaling, this is not a reason for us to request additional data. However, if there are different active sites of the biological or different target receptors involved, which may have a different impact in different indications, additional data are necessary, but not necessarily clinical trial data. It could also be functional assays that are more sensitive usually to detect differences. So I would like to go through some examples for biosimilar products that have been licensed in the EU. For Filgrastim, the concern was the use for stem cell mobilization in healthy donors. But we have to say Filgrastim is a rather, let's say, simple biological. It is very well characterizable and is not glycosylated. It binds to one receptor, and in addition to the data on the stimulation of absolute neutrophil count, we also request data on the stimulation of CD34 positive cell count, which provides additional evidence for extrapolation to the stem cell mobilization indication. For Filgrastim, you know that immunogenicity is not a particular concern, it's very low and has no impact on efficacy or safety. And we do have some post-marketing studies that actually confirm efficacy and safety in all approved indications, including stem cell mobilization in healthy donors. And here I show you some data for a biosimilar Filgrastim, not because I think that's the best one, so I don't want to do advertisement here. It's just that these data are publicly available. And here you can see on the graphs, in the two graphs, stimulation of absolute neutrophil count at 2 dose levels. And you can see that actually the curves for the biosimilar and the reference product are superimposable. And the same is very much true for the CD34 positive cell count. So this is what we really like to see when we assess a biosimilar. Coming to epoetin. Epoetin, of course, is a larger molecule and it's more complex and heavily glycosylated. And the concern here was the use in cancer patients, because these patients with chemotherapy-induced anemia need higher doses than patients with renal anemia. As I said, erythropoietins are complex glycoproteins, but with today's methods, they are well characterizable. The effects are mediated by binding to the same EPO receptor, and we have the same mechanism of action in hematopoiesis, regardless of the cause of anemia. Still, we would like for epoetins to have more efficacy and safety data from a clinical trial in a sensitive model indication. So what is a sensitive model indication? That's an indication that is with regard to the population studied and the endpoint sensitive enough to detect relevant differences between the products. And this turns out to be the population with renal anemia. And also this population carries the risk of purette cell aplasia induced by anti-epoetin antibodies, especially with subcutaneous use. And because we can extrapolate from a high-risk setting for immunogenicity to a low-risk setting, we allow extrapolation of immunogenicity data from the renal anemia to the cancer setting and from subcutaneous use to IV use, but not vice versa. And also here, post-marketing data have confirmed efficacy and safety in all approved indications for the biosimilar epoetins. Again, I'll show you some data from a biosimilar epoetin licensed in the EU. Here you see very similar serum concentrations of the epoetin biosimilar and the reference product at steady state, and also very similar increase in hemoglobin levels. And again, this has been confirmed in an efficacy and safety confirmatory trial.